Assalamualaikum and good morning. My name is Sufi Adha bin Muhammad Noor with matrix number S56133. Today, I want to present my slide proposal with title Meta-Analysis Study on the Detection of Adulterants in Stingless bee honey. Honey is a natural sweetener that has been used widely since ancient time. It is widely used in numerous applications among natural products, including for clinical applications, and it contains about 200 various substances. It also gained more interest and popularity due to the claim that it has more beneficial health effects as compared to the common honey. Research on stingless bee honey have reported its biological properties, which are antioxidant, anti-inflammatory, antibacterial, antiviral, and anti-ulcer activities, and can be utilized as medicines. Adulterants are the main issues in stingless bee honey products because it will affect the original quality of stingless bee honey. Low-cost sugars and commercial syrups are common substances for honey adulteration. Describe well-known adulterants from sugarcane and sugar beet. Several analytical methods have been proposed to detect the presence of adulterants in stingless bee honey, which are infrared spectroscopy, chromatography methods, and nuclear magnetic resonance analysis. The objectives for this study is to compile data and review previous research on the analysis and detection of adulterants in stingless bee honey. Next is to perform the meta-analysis study for the adulterants which are glucose, fructose, sucrose, and HMF contents in stingless bee honey. Lastly is to investigate the relationship between this study and physical-chemical properties for each of adulterants. Next, for problem statement, I noticed that the adulteration process has become a serious issue in stingless bee honey. For methods to detect these issues have been performed, but the effectiveness is not being studied yet. Moreover, the systematic review through meta-analysis regarding the relationship between adulterants with physical-chemical properties is not available yet. For significance of this study, it can lead to future discovery on the development of stingless bee honey as a high-claim health products in the future. It also can improve our knowledge and the beneficial information on the quality control of stingless bee honey. And for material of study, I have reviewed research articles that covers 10-year period from 2011 to 2020 using the online database such as Web of Sciences, Science Direct, and Scopus. My limitation for this study is only using meta-analysis study. Some of analysis has been properly used, such as one-way ANOVA and post-hoc tests. These are the journals that I have been used to search and explain about the properties of stingless bee honey. In the previous research, there were also a lot of study about the chemical constituents and biological activities that related to the stingless bee honey. Based on this research, we suggest a straightforward approach for detecting adulterants in stingless bee honey by combining chemometrics with FTIR ATR spectroscopy. Second, NMR methodology allows all variables to be investigated rather than separately, as is the case with the most often examined parameters. Third, separating and identifying semi-volatile and volatile components in honey is done using the SCMS and GCMS techniques. Meta-analysis study is a systematic review that collects and synthesizes the findings of research that answer a specific topic using a defined systematic methodology. Furthermore, a meta-analysis of effect estimates is a statistical methodology used to synthesize data, creating a quantitative summary of outcomes where study effect values and their variance are known. For material of study, I have used three types of stingless bee honey, which are Heterotrigona itama, Geniotrigona thoracica, and Tetrigona apicalis. And for study parameters, I have divided into three parts of parameter. Firstly, the different species of stingless bee honey. Second, the concentration of adulterants. And lastly, physical-chemical properties that related to adulterants. I have summarized the methodology for this study in this flowchart. For data collection, some of the keywords has been used to propose this study. All these keywords are collected from the research articles, and then it will extract from the sources and implementing into Webplot digitizer software into a graph. Furthermore, Prisma protocol will be used as a guideline to conduct this study. For the process of data collection, I have keyed in some keywords that give me the total number of relevant articles that related to physicochemical data and adulterants in Stingless honey. I have collected the data from Science Direct, Scopus, and Web of Science, and the total number that I found is 8. And for data analysis, all data will be subjected into normalization tests and continued using SPSS software. The method of statistical data analysis, such as one-way ANOVA, will be conducted to evaluate the different values among the collected data. Lastly, the result will be presented in forms of table and figures. First of all, I have collected the data of HMF content in Stingless honey and convert this data into a box plots graph by using web plot digitizer based on the group of countries such as Malaysia, Thailand, Colombia, and so on. Next, for fructose content, I also convert this data into box plots to differentiate the value for each of comparison between the parameters. Furthermore, the glucose content is the one of main parameters that have to be included to detect the physicochemical and adulterants in Stingless honey. Moreover, I also got some data for maltose content from Malaysia and Thailand. The differences of means values and other results are determined by using ART software. Lastly, the total sugar content is also one of the main parameters that has to be included in this research. The result for this parameter also obtained from this graph. For my future works, this systematic review on the status, occurrence, method detection, and analysis of adulterants in Stingless honey is expected to be done. Moreover, this meta-analysis study of raw versus adulterant Stingless honey based on physicochemical data such as HMF and sugar contents.